welcome to the welcome to the Museum seminar today. It's my great pleasure to introduce Haitao Wen from Ohio State University today. Haitao is really a pioneer in immune metabolisms. So Haitao obtained his medical doctorate and Master of Science degree from Shandong University School of Medicine. Then he went to University of Michigan Medical School for his PhD, studying regulation of sepsis. And then he went to University of North Carolina at Chapel Hill for his postdoc work with Jenny Ting. And he focused on regulation of inflammation and extended his work to both how inflammation impacts metabolic disorders and how metabolism regulates inflammation. I think in his own independent career, he really brought us into a full circle, studying immune metabolisms in the immune response. Haitao started as an assistant professor in Chapel Hill, and then he moved to Ohio State about 3 years ago. I think just earlier this year, it's really great to know that Haitao was officially promoted to associate professor with tenure. So congratulations, Haitao. We look forward to your fantastic talk.
Great. First, I want to thank all the organizers. So it's a lot of effort and the patience to organize this so comprehensive immune seminars, and we highly appreciate. Especially, I want to thank them giving me an opportunity to present our recent work. To be honest, I haven't officially given a seminar for almost 1 year, so it's really nice to come back to this formal atmosphere to present our recent work. So today, I want to visit relatively the older story, which we published 3 years ago. But we do have some new progress, and I want to share with you. Like half of will be published, and another half, less than half, will be unpublished data, which I appreciate is any comments come back. So let's give a very brief introduction of what our lab is working on. So as she just mentioned, when I was a graduate student and a postdoc, my training is really focused on the innate immunity, especially with Dr. Jenny Ting at Chapel Hill. So we studied NR biology for about 5 years. So as a innate immune cells, so all the innate sensors, including the toll receptor, AR, RIGI, and the DNA sensing CGAS. So this is a very brief what is the innate immune cells can do. So all this highly evolutionarily reserved, the PR, we call the pathogen recognition receptor, their function is to detect the pathogenic the triggering and the initial inflammatory response. And meanwhile, they also have a counteractive mechanism to control the immune response. Otherwise, there will be autoimmune disease. So, how to move on? Okay, so when I set up a lab about 8 years ago, I gradually diving into this relatively new field. The reason is because in the later phase of my postdoc, I work on some mitochondrial metabolism for the NR, one specific NR molecular, which lead to my independent research. So this is basically biochemistry 101, right? So I'm sure you read it in the when you study the biochemistry in the college. So talking about the cellular metabolism, including several very major pathways, including the glycolysis from glucose. Then after several enzymatic activity, they go to the pyruvates, then into the mitochondria through the PDH. And then including this the TCA cycle, we call the mitochondria metabolic pathway, the carbohydrate acid cycle. And in talking about glycolysis, they have 3 major pathways. Glycolysis is one major one, but they do have a pentose pathway. And also another, we call the minor glycolysis pathways, hydroxyamide biosis pathway. So although we call it minor, but actually the function is quite powerful based on our work and also other the lab's work. So the enzyme is called the O GlcNAc transferase. The function of this enzyme basically mediates the protein modification. It's called O GlcNAc. So it's a sugar moiety modified serine and threonine residue and regulate the multiple function of the targeted protein, including the protein stability and kinase function and motility, and also the transcript factor function, because the target is including multiple types of the proteins. So our basically research, basically on several years ago, we began to investigate what's function of O GlcNAc transferase in the immune system. Meanwhile, we do have another red chain in the mitochondrial pathway, but today I only focus on the O GlcNAc transferase pathway. Yeah, so it's not moving well for this. Hold on a second. Let me close this. All right. So as a metabolic field, so the how you visualize the field, right? So when I started to visualize the field, basically I want to emphasize based on the evolution of the system. So the nutrition utilization and also the immunity is a two while evolution highly conserved system. They evolve together. So that's the reason we call the interaction between metabolic reprogramming and the immune activation is very, very intense in multiple layers. So it's like a two-way the directions. So let's take a one way. So basically the immune activation can cause metabolic reprogramming. So this is well established because you activate the macrophage, the nuclear cells, you cause the dramatic changes in metabolic reprogramming, including the enhanced glycolysis, Warburg effect. Meanwhile, they shut down the mitochondria respiration. And so how can you do that, right? So how they do that? That's kind of, it's like a very intense investigation, and what's the immune activation cause metabolic reprogramming. So there's a multiple mechanism, including the transcriptional, translational regulation of enzyme. Relocation. So we talk about metabolic change. You talk about the enzyme and enzyme move and enzyme change, and also the affecting activity of metabolic enzyme and the others. But meanwhile, another important the direction we want to understand what's metabolic the reprogramming do to the immune cells, because that's why we study the immunometabolism. That's how you get the sense of can we use the metabolic reprogramming to use as a therapeutic therapies to treat the immune-related disease. So there's several mechanisms how the metabolic reprogramming can affect immune activation. So most importantly, in my view or point, I think is the metabolic immediate post-translational modification, including acetylation. This is used acyl-CoA to do the lysine the modification. O GlcNAc, which is our research target. Meanwhile, recently there's many different the PTM, including palmitation, succination, ADP replacement, and the lactation. This is all come out. So it's quite a comprehensive the modification of proteins. Meanwhile, they also have the metabolic, the metabolite affecting the activity of immune, the enzyme. For example, the pioneer work by Dr. Luke O'Neill, they published the first one in the succinate and the PDH pathway can affect the interleukin-1 beta production. And RAS and RLS and IS derived from mitochondrial metabolism can affect these metabolic reactions. So another one is IDH mediate the epigenetic change can regulate the protein regulation in the macrophage. That's another the intense the investigation field there is movement. So I think from here our overall the mechanism the goal of immunometabolism lab, basically we want to investigate the intrinsic crosstalk between immunity and the metabolic networks. And then we identify targets, and finally we design a small molecule to treat the therapeutics, immune disease. So the pathway we are interested in is called hypothalamic biosis pathway. Actually, is branching out from this F6P. The entry enzyme is called GFAT. So this is an entry enzyme to control the flux of these pathways. So the downstream, there's an important enzyme called O GlcNAc transferase. It uses UDP GlcNAc to do the PTM, post-transcript modification. So overall, from the history, the overall the mechanism, how this OGT, the PTM works. So since this protein oxidation targeting on the serine and serine residue, it's almost sometimes it's the same as a phosphorylation site, sometimes adjacent to the phosphorylation site. So the historical viewpoint, the protein oxidation is a counteract to phosphorylation. You can imagine how important is the protein phosphorylation regulating the immune function, right? So that's how the OGT works. But our data, there's many the data from different labs just suggests this oxidation is modulated more than just the counteractive response to the phosphorylation. It also can regulate the protein stability and the kinase phosphorylation and the transcription factor activity. But I want to remember, so this is how the OGT mediated protein PTM works. So from the history, so O GlcNAc protein, the oxidation has been studied in the literature for more than 40 years. It started from Dr. Hart and in very old biochemistry, old school studies. From there, so there are many different the aspects has been identified for OGT mediated the functional. So generally speaking, OGT mediated protein oxidation is a very fundamental activity towards regulating the almost all aspects of biological activities. So we are in the signaling pathway, in signaling transduction, development, and the importance of this development story, because if you remove the OGT in the whole body level, the embryo just died in a very early embryogenic the stage. And the cancer, metabolic disease, any disease, you can think about it. So the mechanism I just mentioned is through the regulate crosstalk with phosphorylation. Especially in the immune system, people have identified several important oxidation targets, including the toll-like receptor pathway and the AFKB pathway. But until now, so there's an overall function of OGT hasn't been identified in the macrophage yet. So this is another one OGT function in the cancer, which we are working on, but today I will not discuss OGT cancer. So since today we are going to involve the RNA sensitive pathway, RIG-I and the MAVS mediated RNA sensitive pathway. So I'll give a very brief introduction of what's the nucleotide RNA species detection system. So this is identified by 4 different groups years ago. And the MAVS, so this is important, the adaptive protein in the outer membrane of mitochondria. So the real sensor of the RNA species is called RIG-I or MD5. So this is has been the crystal structure has been solved to see the real binding between the 5-triphosphates, the double-stranded RNA binding to the grooves of the RIG-I. That's also been identified years ago. So the function of this pathway RIG-I and the MAVS pathway is generating, activating this IKK epsilon TBQI pathway induced type I interferon, and as well as AFKB dependent inflammatory cytokine pathway. Then this most of the we call the ISG interferon stimulated gene. The function of ISG is counteract the viral infection in multiple steps of viral infection. So since this paper already been published, so I'll just give several slides to pick up the highlights, the essence of this paper. So overall, this paper identified, actually the Tianliang Li is the first author in this paper, and he identified, so the OGT can moderate this MAVS adaptive protein. So this modification of MAVS is a required step for the downstream the MAVS ubiquitination, as well as a high complex formation of MAVS. And the multiple protein complex formation can lead to downstream the FKB pathway. If you remove this OGT or you remove this MAVS organization sites, which is serine 366, the downstream activation will be defect, and as well as impregnation of MAVS. So this is overall of that 2018 paper. So after this paper, there are several groups found a relatively similar finding of on MAVS modification. So these two identify this the OGT deletion can cause defective RIG-I and MAVS induced type interferon production. But this one used influenza. Actually, influenza project is a little bit different, which I will introduce later, because in vivo data will be updates depending on how the information it's contribute to pathogenesis or fighting against the viral infection. So this is OGT on the HCV pathway. And the last very recent two papers, they talk about OGT HPP pathway. Overall, generally one sentence of OGT function in antiviral immunity is if you remove OGT, if you decrease OGT enzymatic activity, you enhance viral implication, the pathogenesis of viral replication. So that's put OGT as an important metabolic, the niche for antiviral response. But that's not just one simple story. So after we published one, we also realized there are several important questions we haven't identified yet. So first one is what is upstream of OGT? And we delete OGT, right? So that's easy for immunologists to study, but what's in the real situation, what happened to, what regulates the OGT and the activity? Does it move? So this is several important questions we haven't addressed yet. Is this real, the OGT antiviral activity? It really depends on just the MAVS, type interferon, ISG pathway exclusively, or there's other activity involved in the OGT. This is another important. So basically, we studied the MAVS and RIG-I pathway. They think, okay, automatically this is dependent on the real, but sometimes if you really look at it in details, you can find a very surprising the results, which I'm going to show later. And also the different function of OGT different cell types. So this is the three questions we want to answer after we publish the first one. So very brief introduction of this, because we have to, based on the old publication, then get into the new development. So in this the 2018 paper, we found firstly we performed the metabolic assays because we want to look at what happened to, in these different pathways after viral infection. So we use VSV, which is very well established the RIG-I MAV dependent and virus model to stimulate this RNA virus pathway. We found that the VSV infection for 6 hours can cause profound profiling change of metabolic metabolite. So this is just some metabolic studies. Then we look at the individual metabolic pathways. We realized that this viral infection actually can enhance this glycolysis pathway, 3 of them. So this glycolysis pathway look at different metabolites. PVP pathway, yes. And the interesting for HPP pathway is also increased. So this first glycolysis and PVP pathway, that's already been published. I will not feel surprised. I did not feel surprised because this is the old story based on the 10 years ago, published by another group. But the HPV pathway is quite surprising to us. So this is one snapshot of metabolic profiling, right? But we have to understand, when you study the metabolic activity, you have to understand the metabolism activity is a flux. It's not just the one snapshot. It's like autophagy, right? So get in, get out, get in, get out, right? So you have to understand, you look at the individual time points, that's snapshot. So you think one metabolite go up and down, you do not know the flux. To understand the flux, you have to use a Chesney assay.
So this is what we do. We put on the carbon 13 labeled the glucose, then look at the downstream of UDP GlcNAc labeled by carbon 13. It is increased, consistent with the snapshot metabolic. So that one we know, okay, this pathway is enhanced. Then the very natural question, well, if you remove this pathway, oh, hold on a second. Before that, I want to know this pathway activity is increased, then what's the downstream function, right? So basically it's a OGT-mediated protein oxidation. Luckily, due to the early work by the the Hart, they have antibody, which is really lucky for the oxidation field, because most of the metabolite, media metabolite, the modification, there's no antibody detect. This has antibody. So we found that after the several hours of VSV infection, another stimuli used is poly-IC. As similar, and the data enhanced in the early time point, the later time point is dropped down. So this is also very predictable, because the OGT, the oxidation, the activity is always combined by the OGA activity. They are not separate. They are always together. One pathway, one enzyme activity goes up, they automatically induce another pathway goes up. So that's the reason the oxidation, if you look at the oxidation in the cellular level, it's always dynamic. So we have to keep that in mind. So that's the reason we need to pick up choose a different time point. But at this 6 hours, this increase. So then the natural question, what's the functional relevance, right? So very simple, as we generate this conditional knockouts by cross the OGT flux with the lysosome create. This is the myeloid specific knockout. And interestingly, we found that with deletion of the OGT in the macrophage, then the cell culture based assay, we found the virus induced the interferon and also the ISG interferon stimulated gene, including the ISG15, IP10, and MX1, all attenuated in the OGT deficient macrophage. So that's give the answer. The OGT is critical for the RIGI and the MAVS induced pathway antiviral response. So this is the mRNA level, and this is the protein level. So this is RIGI and the MAVS pathway. So another parallel pathway will be, how about the DNA sensing, which is C-gastin pathway. We did that as a control, and we use poly-DADT stimulation, the interferon stimulating DNA species, as well as direct stain agonist, the CGAMP. We found that in this specific bone marrow-derived macrophage, deletion of OGT caused no phenotype in the C-gastin-induced pathway. So this is specifically to the RIGI and the MAVS pathway induced the terpene interferon production. So RA level and the protein level. And when we look at, then this is the transcription. How about the signaling pathway, signaling transduction pathway? So we give VSV stimulation, and we look at the different signaling molecular underneath this in this pathway, including the TPK1, phospho-TPK1, AQK epsilon. So this is a TPK1 epsilon pathway, and the STAT one will be the responsive to the terpene interferon is all decreased in the GlcNAc. So this is consistent with the cytokine, the RNA data, and the ELISA data. We also look at the FCARB pathway as also attenuated when we removed OGT in response to VSV stimulation, and same as the phospho-ERK, so one of the Mechanistic pathway. So far the data is consistent. If you remove OGT, the responsiveness to VSV induced the RIGI and the MAVS pathway is attenuated. So animal model shows consistent data, because if you don't have the effective antiviral terpene interferon pathway, then you cannot efficiently control the viral replication in the lung. So this is a VSV, the IP model, which also being used in literature quite intensively. So we found if we remove OGT, specifically deletion in macrophage, in myeloid cells, it causes more the animal deaths, and the antiviral inflammatory response can decrease. And but meanwhile, the viral replication can enhanced due to the. At this paper, we said it's due to the attenuated terpene interferon production because it's combined together, right? But later on, I'm going to show the data. It's not the whole picture of OGT-mediated antiviral response. But so far, the in vitro and the in vivo are consistent. So next, we want to look at the more details into this enzyme. So OGT is a very, very interesting enzyme. It's big. So they have N-terminal 13 TPR repeats. So usually in the literature, in the literature, TPR basically domain is mediated protein-protein interaction. So this is kind of well established. This is how the OGT can recognize its target. And in C-terminal, they have two catalytic domain. It's called CD1, catalytic domain 1 and 2. So that domain 1 basically mediates the protein modification by the UDP GlcNAc. So we ask question: is this antiviral response depend on the enzymatic activity? So to do that, we have generated the loss of enzyme mutation of OGT. So one of the key sites is lysine 908, replaced by the adenine. We found is lost the entire the O GlcNAc transferase activity compared to the wild-type OGT. So we generate this the viral vector mediated the reconstitution the experiment. When we put we compare the wild type to knock out the cells, the terpene interferon getting decreased. But we put back, reconstitute this knockout with either wild type or OGT, the loss of mutation OGT. We found the wild type definitely can rescue this attenuated terpene interferon production, but not the K908, the alanine mutation. So that's tell us the OGT mediated antiviral response requires its enzyme activity, because if you remove it, you cannot rescue the terpene interferon production. Same as the downstream, the signaling pathway, including this phosphor earth, because if the phosphor earth only rescued by the reconstitution by wild-type OGT, not by the loss of enzyme mutant of OGT. So this is very consistent with this enzyme activity is important. So next we begin to identify what's the target of OGT. So one of the common strategy for innate immunologists to look at signaling pathway, we do the luciferase assay, because that's we can reconstitute the different the protein at the different levels. We can test one by one. So when we put the RIGA MD5 MAVS together with OGT, we found OGT can enhance. However, if we put the downstream ERF3 active epsilon, they cannot. So basically that tells us OGT target on the MAVS or the higher than MAVS, right? So this is kind of how the test, like a semi-unbiased screening to test how this enzyme works in different levels. So then we look at if the OGT can modify this protein. We found the RIGA and MD5, there's no modification. However, if we look at the MAVS, so we found a very strong modification of the MAVS by OGT. So baseline MAVS, they can be modified in the basal level. But if we the co-expression of OGT with MAVS is significantly enhanced, so the signal is dramatically increased, but not by the loss of enzyme activity OGT. So this is again test the OGT function requires enzyme activity. Finally, we look at the endogenous of MAVS. We co-IP we immunoprecipitate the MAVS, then detected by the antibody. We found the virus can enhance the endogenous MAVS, the oxidation, but not in the knockout. So this kind of tells us the OGT is important to modify this MAVS, both in the expression or expression system as well as in the endogenous system. Then we do the mass spec by multiple rounds, then followed by cytodirect mutagenesis. We found one key, the sites, serine 366 is important for MAVS modification by the OGT. So finally, we mutate this site, because the point is we know this site important. Then if you remove this site, is this related to the functional reconstitution for the type interferon response, right? So we use a wild type MAVS to in response to VSV, we can induce the type interferon production. But this data is generated in the MAVS knockouts fibroblast. So the VSV cannot induce any type of interferon in MAVS knockout. It's really depend on the regard MAVS pathway. So this is a good because we already we show the MAVS important. But when we reconstitute the MAVS knockout fibroblast with wild type MAVS, but not with this loss of mutation MAVS, we can show the only wild type MAVS can induce the type interferon production in response to VSV. So that tells us this site, the 366, the modification is important for MAVS function. Okay, finally, we doing the more biochemistry work, and we found this the 366, the modification is important for MAVS, the ubiquitination, because we co-express the HA, the ubiquitin, together with OGT and MAVS. So OGT can enhance this MAVS, the ubiquitination, and in K63 the format, not in the K48 format. And we next try this is a K63 only ubiquitin, and it's not, we also try the K48 only ubiquitination. It's not working, but it works in the K63, the mutation. And when we look at endogenous, this is a K63 ubiquitination based on the MAVS ubiquitination, which is consistent with the notion the K63 ubiquitination is very important for MAVS secondary transduction and the antiviral immune response. And so this modification is depend on this ubiquitination, because when we compare the wild type OGT and the loss of the enzyme, the mutant of OGT, we found the wild type can enhance the K63, but not by the loss of function mutant. And in another layer, so when we remove this series 366, the sites, the loss of the ubiquitination, but meanwhile they also lost the K63 ubiquitination. So therefore we confirmed this OGT mediated the MAVS ubiquitination, both functional in OGT level as well as in MAVS level. So this is very important in the OGT field. We want to propose some function protein is modified and is functional. They have to test both there. So to finish this entire circle. Okay, so this is what we published. Okay, and then like I said, I want to move on to something new. So before I do that, so I will give a very brief of this influenza virus. So the one we done is in the VSV, which is typical regard MAVS pathway. And if you have attenuated antiviral the interferon pathway, you induce the more pathology in response to the VSV induced pneumonia and the mortality. So that is a textbook. So every lab can recapitulate that response. Influenza is a little bit different the story, because based on the influenza pandemic, it's almost 1 century ago. So majority of the influenza-induced mortality, not because of you have less information, but rather a majority of the patients, as well as animal models show, the mortality is due to the enhanced inflammatory response. So something like a SARS-CoV-2. So because it just induced too much the side effect in the pathology in the lung. So basically you just kill the lung. So, but if you talk about virology of influenza, when influenza gets in, so usually the RNA will go to the nucleus, and here the induced mRNA synthesis. And then the mRNA synthesis will be detected by the RIG-I and the MAVS pathway. But this is not a complete story, because recent papers show the mRNA stasis, as well as some we call it defective the genomic RNA of influenza, also detected by another RNA stasis. It's called the ZBP1. So that can induce the cell death. But this RIG-I and the ZBP1 RIP3 pathway, they are not redundant, because RIG-I MAVS pathway is mostly focused on type interferon production. But the RIP1 and the RIP3 pathway mostly is the induced cell death. So it's different. Today, I'm going to talk about there are something new, not just about type interferon and not about just virus-induced cell death, but rather they have metabolic related activity, which is kind of a really unknown field for the antiviral immune response, and linked by this OGT biology. All right, so why don't you move? Okay, so to start that, we generate the new model system. So we found I already showed this piece of data, right? So licin 908 mutation loss of enzyme activity. Actually, there's a previous paper almost 10 years ago. They identified several of them. So why is important for this licin 908? So this one actually is the crystal structure is here. In that paper, they call this 898. Actually, it's the same as the licin 908. It's just different calculation of amino acid. The function of this licin actually is to capture the substrate, not to target the substrate, UDP GlcNAc. So they capture UDP GlcNAc, then modify this target on the serine stranding residue. So this is quite important. If you lost this the site, then you cannot grab the substrate. There's no modification, right? You lost activity. So this is a very established the model system. So we begin to address, is this really enzymatic dependent? The reason because there's several, not big paper, there are several papers. They mentioned about this N-terminal of TPR domain. They have the scaffold protein function. This protein is big, right? So it will not surprise me. The scaffold protein will have some the non-enzymatic activity function. The idea actually is also come from necroptosis pathway. Because if you know this field, the necroptosis field, there are many functions of kinase, RIP1, RIP3. They are not dependent on the enzyme. Actually, it's really dependent on the scaffold protein function. So that is really beyond the people's, not beyond the people's imagination. People can imagine that, but it's just really surprising me. So to reach the one of the solid conclusion, you have this model system to test it. Okay, so this is what we did. So we generated this inducible knocking system. So short of this is quite the difficult to answer. But the long story short, basically, if we have 2 LAX-P here. So if you use Cree to remove this LAX-P, the ending product basically is end up here, the 908 alanine. Because you removed this endogenous, the Y-type site, you end up here. So if you don't have Cree, this is basically one of the Y-type OGT, because this site is here cannot be expressed because they have stop codon here. Okay, but if you remove, basically you just loss of the enzyme X. Here is it come up. So if you use Cree, yes, this is you can induce the loss of enzyme activity. Initially, we saw that this enzyme activity actually is quite important, but it turns out when we use the E2E Cree to generate the whole body knock-in system loss of enzyme activity, actually we surprisingly found we can get live mice. So this is after the 2 rounds of the OGT knock-in and by the OGT the flux knock-in flux to E2E Cree to generate the whole body knock-in. Initially, okay, maybe that's not our model system is not working. It turns out our model system does work. Look at this one. Look at the heart. So this is the whole body knock-in system. Okay, of course we have other cell type specific knock-in, but if you look at the whole body knock-in, So wild type knockout in heart, liver, it's just too much protein, and this is bad immunoblood. Never mind all this one. In spleen, in lung, and in the intestine, so they are all decreased in the protein population. So it seems like the model system works. So since we have this mice, then we generate the MAVS. So next we want to look at just to repeat what we have done for the anti-VSV immune response. So now surprisingly, when we affect this O GlcNAc MAVS, so we found the viral replication is enhanced. So this is kind of consistent with what we shown in the antiviral response is decreased in the O GlcNAc, right? So if you lost enzyme activity, then the OGT, the viral replication is enhanced. This is by the NP-mRNA, the mRNA, and the S1 mRNA. Is this reflect the viral replication? Yes, we use the GLP PR8 is also increased. We also did the titering, the plaque assay. We found the same level of the virus in the supernatant released by the NAC the MAVS. Every time we found it's like a double the number compared to the wild type. It's another way to look at the viral replication. We look at the individual the proteins infected by the PR8 influenza. So it's also enhanced in the NAC cells compared to the OGT flux the fibroblast. This is due to the different infectivity. Actually, it's not. When we look at the genomic RNA, we found the genomic RNA between the wild type and the loss of enzyme activity, the MAVS is similar. So basically, the cells can take the same amount of virus. It's just the virus transcription and the replication is different. It's enhanced in the NAC in the cells. Then we did quite a bit of the extensive studies. We use DERT S1 PR8s, and we use Sendai virus. We use VSV. We use VSV pseudotype with Ebola gastrin protein. Finally, we also tried the SARS-CoV-2 for 2 times. And this is all the collaboration between Shan Liu and Jacob Young in our at OSU. So it's a very good teamwork. So we found that in all these cells we tested, the NAC the loss of enzyme caused the enhanced virus replication. We also tried the NAC macrophage. So this is lysosomal based on NAC specific in myeloid cells. It's also enhanced. Okay, so it's very consistent with our previous finding, right? So you have less type 1 less the enzyme activity, then the less antiviral response, then you have enhanced viral replication. So everything perfect. Is this really due to the type 1 interferon? So this is a question, I know if we want that question, maybe you can come out different answers. Actually, this is what we found. So next, to test if this is a defective type 1 interferon production, is this responsible? So we have to do the double knockout, right? So we remove the RIG-I, MAVS, and ERF3. So in the wild type, it's double like enhanced. If you remove RIG-I, MAVS, and ERF3, just look at wild type. Yes, this pathway is important for antiviral replication because the viral replication is enhanced in the RIG-I knockout the system, in the MAVS knockout system, actually in the highest in the phenotype in the ERF3. So this is not surprising because so the ERF3 control almost all the ISG. The RIG-I and the MAVS is just one pathway to regulate. There are several others like a PKR or some OAS pathway. We haven't focused on that one, but nonetheless, we remove all these pathways, we found the phenotype still there. So this is the PA mRNA, this S1 and MP mRNA. So this is mRNA. So this is we show the CRISPR-based strategy works. RIG-I knockout, MAVS knockout, ERF3 knockout, the functional. So this is quite surprising to us, right? So it seems told us that OGT immediate some RIGA and the MAVS independent antiviral response, which may be immunology, maybe not immunology, right? So that's what we need to figure out what. So since this OGT is an enzymatic activity, it's an enzyme, so we go back to our long-term collaboration on the metabolomics. So long story short, so basically we performed this metabolic assay. We have the preliminary evidence suggests this metabolic pathway in the fatty acid metabolic pathway. Some intermediate metabolites is changed. I won't put this, there's a long list there. So this very early stage, I haven't, this project is not ready to publish yet. So the long story short, we found the fatty acid metabolism is changed between wild type and knockout. So very brief, the introduction of what's a fatty acid metabolism. So fatty acid metabolism including both synthesis pathway and degradation pathway, right? Synthesis pathway, there are like two major branches. First one is cholesterol synthesis pathway. Another famous name, you probably know the name, is called MAVS pathway. And the key enzyme is HMG-CoA reductase. And because this is a generate cholesterol, is very important for heart disease, right? So there are many different studying. The drug has been functional in clinic. It targeted on this pathway, MAVS pathway. And another is fatty acid de novo genesis pathway. So acid-CoA, they go to the different enzyme activity. Then finally, they go to the triglycerides and go into the triglycerides and store this fatty the component as a lipid droplet. It's called LD. So this is a synthesis pathway. What about the degradation pathway? Degradation pathway is simple. It's just called the mitochondria beta oxidation. Use the CPT1A and the CPT2 transporter system to chop off the carbon. Every cycle, they have like a 2 carbon decrease. The long chain going to boom, 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 then go to the short chain. Use the fatty acid, the free fatty acid in the cytosol. Synthesis, degradation. So what's important, all right? So far, we begin to test each of them. So we remove the CPT1A. They basically first we want to look at the fatty acid, the beta oxidation FAO pathway. So we remove CPT1A, CPT2. So this is CRISPR deletion. Interestingly, this beta oxidation is important. It do play a role in the antiviral immune response. It just not play a role in OGT system. So because we remove CPT1 and CPT2, the phenotype is still there. But if you just look at wild type, yes, this pathway is important. So why it's important, but it's another project, right? So it's not related to the OGT. And then we look at the cholesterol synthesis pathway here. So this one we use a chemical because the chemical is well established in the system. We use 2 statin, simvastatin and cerevastatin, and we can abolish. And then we also add this intermediate metabolite called the malonate acid. Actually, the malonate lactone is a precursor of malonate acid. Then basically you want to enrich this pathway. But if you enrich it, it's still not remove this phenotype between this wild type and the knockout system. So it looks like cholesterol pathway and the beta oxidation pathway is not important. So finally, we move to the fatty acid they call it de novo lipogenesis pathway. So this is 2 chemical we use. So far, we only have the solid data for chemical. We do have preliminary data on the genetic, but it's too premature. So I don't want to confuse here. So the TOEFL is first one is ACC. Actually, I want to show you where the ACC is here. And the C75 is an inhibitor of next one, FAS. So we found this one works. So, okay, great. So we know the OGT controls some lipid dehydrogenase pathway to control the antiviral immune response. So you may feel, wow, this is boring, right? So lipid metabolism and lipid droplet has been published more than, if you check PubMed, it's more than 200 papers, including some cell paper. They're important for virus replication, including HCV, and some polio. But in our story, we more focus on what's upstream, right? So we know OGT is important. So this is the first time we show the OGT can link this immunity to metabolism. I think that's our selling point. Of course, how the OGT upstream is still a mystery to us. We do have several directions we want to go, but at this moment, this project is too early to tell for sure. Okay, so for this new progress, I do not have a take-home message because this is quite premature, but it clearly gives us some direction to move forward. So I consider as I published the preliminary data. Okay, so the 2018 paper is carried out by Tianliang Li, and now he's a professor in Shandong Normal University. So so he is genius. Did quite of extensive genetic work and all the biochemistry work. It's beautiful work by Dr. Li. So I wish him best luck for his independent lab. So we have quite a bit of collaboration inside OSU, Shanlu and Jacob. And also we have quite intensive collaboration outside, including the Dagreen. Chengjiang is a very helpful the collaborator on the MAVS studies and the different mutations. So they have different funding from GMS and NIAID. So I will stop here. I didn't see the time. What time is it? Oh, okay.
The time is perfect.
Perfect, yeah. Perfect.
Really nice talk, Haitao. It's really, really deep mechanism understanding about how OGT works. We already have quite a few questions lined up. Guangxuan, Meng, you can go first.
Hi, Haitao.
Yes. Hi. How are you?
Very nice to meet you online. I heard your name quite often. Actually, the work you introduced, I heard about 2 years ago when I was attending a meeting of Keystone meeting in Taipei, chatting with Jenny Ting. I'm Guang Xunmeng from Institute Past of Shanghai. Chengjiang and Tianliang, these are all familiar names in China.
Sure.
Okay, so your work is very impressive. I just have a brief question for the first part, the mouse part. You mentioned the ubiquitination is affected by the OGT modification. How about the aggregation? We know the mouse signaling needs to form aggregates.
Of course.
Did you check that in your experiment?
Definitely we want to do that. It's kind of a natural question, right? So ubiquitination important for based on the Chengjiang's work, and we tried a couple of times. Not I have to say, not very hard. We tried a couple of times, and that's Joe the aggregation assay. It's just it's not a very, we call it PQ, right? Printer qualification. It's not there yet. So that's the reason we didn't use the data. But our hypothesis is that it will affect the MAVS, the high molecular organic, the oligomerization part. Yes.
Yeah, a related question is, if the OGT modification can increase the ubiquitination, do you think in general different forms?
You're frozen.
I think Guangxuan may be frozen.
Yeah, he's frozen.
Yeah, we can come back to Guangxuan when he's back online. Xiaolei, you can go next.
Okay, hey, Haitao, this is a really beautiful work and very nice mechanistic insights, I think, not only on the OGT, but also on the scaffolding function. So maybe I can follow like one of the questions that Guangxun just have. So in the catalytic dead mutant, so do you find actually like that one also affects like for example, the MAVS aggregation or right? In other words, does MAVS have direct interaction with like the TPR region of the OGT?
If the OGT can direct the interact with the MAVS, so we found so we didn't do that the direct experiment. So we just we assume that OGT MAVS because it's a direct target, right? So I will say the interaction is there. It just but the more important is OGT modify the MAVS. So that's the reason but I probably will do it because later on we have now the new the cells I want to test is if OGT moves, can we directly go to the mitochondria mediated by MAVS? So that's a very good suggestion.
Yeah, because like I think a general question will be like what determines the specificity of OGT, because it seems there's no let's say a motif that the OGT can recognize on substrate. Right. It rather depends on the interaction of the TPR and between the TPR and the substrate. Right, if I understand correctly, right?
Yes, that's true for now, because you know there are several OGT labs. I think this question is very obvious, right? Everybody wants to understand how is there any like a generalized mechanism. Mm-hmm. So far, there's no like you know, you can publish this, publish that, but
so I guess it's context dependent.
Yeah. Right.
Okay, good. The other question is, so in your 908 knocking mouse, did you find the OGA expression level is affected or not?
Very good question. So I haven't tested the OGA level yet. So that's very, I won't be surprised to see OGA also going down, because I think it's 14 years ago, 2006, that the GBC paper, they induced by the the tamoxifen induced the ER3. If you remove OGT, the OGA will drop down after 24 hours. So it's very consistent between each other. So I won't feel surprised too.
Okay, that's great. Maybe the last question: have you looked at the cell localization of the knocking one? Is it similar to wild type or it's different?
Cellular localization
of the 908.
Oh, you mean yes? So the OGT, the localization. We haven't tested the 908 localization. Actually, we do have the confocal data. But we don't know the answer for sure. But one thing we know, the OGT, the protein level is enhanced.
Protein level is enhanced. Okay.
Yeah. So it's that's kind of tell us this enzyme activity actually, yes, you know, use this protein to. But if you remove the enzyme, the protein accumulates. It kind of try to rescue the function.
Okay, that's great.
Yeah, it's a very cool question, because later we have lined up of several assays we want to do. Definitely your question will be some of them already on our to-do list.
Okay, great. Yeah, I think that there are a lot of like interesting things to explore. Okay, thanks so much.
Yeah, Guangxuan is back online. Guangxuan, do you want to just ask your follow-up question?
Yeah,
hello.
I typed it, but anyway, if you can hear me, I can just
I can hear you.
Yeah, briefly ask a question. I was trying to ask the OGT modification, if it increase the ubiquitination, the different forms of the post-translational modification, may they have some sort of collaboration through some mechanism?
in our published story, we found the OGT oxidation actually mediates this TRIM mediated ubiquitination. So it's increased the TRIM and the MAVS interaction.
Oh, it's through trim. Okay.
I think that's just one of the mechanism. They probably have some more even involving the DUBA, the deubiquitination may be also involved. But in our study, we just found that the TRIM, the interaction is enhanced.
Great. Great. Thank you.
Yeah.
Thanks a lot.
Yeah. Thank you.
And next question comes from Wu Chuan.
Yeah.
Hey, Haitao.
Hey.
Nice talk. I just have some general questions. So, first of all, the OGT expression is specific for macrophage, or it's actually also express other immune subtype?
It's expressed by every cell type.
Yeah, I figured.
Cells.
So basically, as as I understand that the OGT should actually, the transferase should function through O-link sugar to the protein. So one of the question is, instead of, or let's say, besides the MAVS, is there any target in the macrophage the OGT actually function upon?
Yeah, so FCARB is another one, and in the target published by another group is I think it's some people published P65, and actually the tab 2, if you know the name, the tech 1 and tab 2. Yeah, tab 2 actually is a kind of a positive control when we do this in vitro oxidation assay. So tab 2 also being targeted by the OGT. Yeah, go ahead.
No, I just say, but from the previous findings, the OGT modification also leads to the ubiquitination, or it's only specific for MAVS?
You mean general ubiquitination?
Yeah.
You know what? For the ubiquitination related the phenotype, I cannot recall any others. So protein stability is yeah,
exactly.
So yeah, probably the ubiquitination.
So
also
that's actually I'm actually quite surprised to see the result, because you normally, no matter it's unlink or O-link sugar transferase. So what's the consequence or the result? Usually it's decorate the different sugars on top of the protein, which actually protect the protein from the ubiquitination or the degradation. Your result actually suggests the otherwise. Your result actually facilitated.
Right.
Is there any specific feature from the MAVS which different from the other protein?
I think it really depends on the ubiquitinase, E3 ubiquitinase. So it really depends on the enhanced ubiquitination or less ubiquitination. It's just the interaction between this target protein and the E3 ubiquitinase, right? So this study realized that it's enhanced the TRIM and interaction with MAVS. So that's the reason we cause the enhanced the ubiquitination. I understand that the CMYC and beta-catenin actually is update. That is related to K40, but I think that paper, they didn't show the ubiquitination. They just see the stability is increased. They didn't do the K48 or K63.
No, but
just assume, right?
Right. I mean, but the reason I'm asking this is because this E3 ligase normally actually interact with a lot of different proteins, not only the MAVS. So that's why I'm curious that why the MAVS actually leads to the opposite consequence compared with other protein decorated with the O-link sugars. You know, that's actually, I think that's quite significant findings, although you know, the machinery probably is somehow unknown, but yeah. So my last thing is actually a rather comment. So have you the OGT actually you're actually look at is particular on the ERs. Did you actually look at also the GOGs?
ER, it's not in the ER. So this is in the mitochondria.
Oh yeah, mitochondria. Sorry. I'm just asking you that if you have other look at other cellular components like the GOG.
Yeah. So we at this moment, since this is related to the antiviral, so we look at we use a viral infection model. We look at the mitochondria. We also have another target organelle. We haven't showed the positive data yet related to the lipid metabolism. So in our follow-up story, we found the replication 3 is also targeted by the OGT, but that's we didn't talk about the organelle. This one is just in the cytosol.
Okay.
Because the nucleosome, well, if you think about the nucleosome, is like a multiple complex, protein complex. So I guess this can be localization. Yeah. So the OGT, the migration is a very interesting topic for us now, but it's too premature to say anything about it.
Okay. Thank you. Thank you.
Yeah. Thank you.
Right, I have a couple more questions. One question is a little more general question too. You talk about the different metabolic influx, outflux. Do you know anything about which process may regulate OGT activities?
So the general answer to that question is it's just the availability of a substrate. And
So it's on constantly.
Yeah. So is any mechanism regulating this enzyme activity? For example, OGT has been modified by OGT themselves. OGT actually itself is oxygenated, but that direction haven't been go too far. We also know OGT is being some E3 glycase being ubiquitinated. And also we haven't go that too far. So upstream of OGT enzyme activity is a very interesting topic. We only know it's upregulated by NERV2 transfer factor. But protein level, the enzyme activity level, not there yet.
Cool. And I remember Xiaoyong was looking a while ago about potentially OGT maybe getting to the nucleus, regulating expression directly.
Yeah.
And in your this really kind of that model, it sounds like a very cool way to study if that's actually the case. Maybe participating into assembly of some of the either silencing or activating complexes.
Right. So by the confocal assay and by biochemistry, the organelle differentiate assay, OGT, majority of OGT is actually localized in the nucleus. So associated with TAT2 or TAT1, that's published by Xiaochun Yu years ago to regulate the some metabolic gene expression. But you know, OGT definitely is not only functional in the nucleus; they also function in the outside. People also published in the mitochondria. It's called O GlcNAc. It's really hard to pronounce. Basically, it's protein of the oxidation. So people isolate the mitochondria, look at the proteinome of mitochondria. So it's everywhere.
That's fascinating. And in the band you shows the modification band on the gels. Are any of those bands nuclear proteins or they're primarily cytosolic proteins? They're targets.
Yeah, I think there are many nuclear proteins being targeted there. So
I see.
We just haven't looked at each of them yet. The Xiaoyong did several MS assay, pulled down OGT or pulled down by anti-organic antibody, then look at ribosome protein, of course, because every single MS, the top list, there will be ribosome protein, right? And some nuclear protein, and some, cycling, yeah, I remember Xiaoyong's data. It's a long list.
That's really cool. Thank you. Yeah,
Right.
I think if there's no more question, let's thank you, Haitao, again for this wonderful talk and discussion.
Thank you, appreciate it.
So hope everyone have a nice the rest of the week.
Great, and we have Dr. Dong will be present this week too, right? Yeah.
Yeah.
And we'll.
All right.
All right, great, and thank
See you
you
guys.
for the opportunity. See you.
Good night.
Good night.